Please have a seat, please. Good evening. I am Dr. Imad Baselius, Chairman of National Organization of Drug Control and Research, one of organization of EDA, Egyptian Drug Authority. I am pleased to be here in this well-established conference, first national conference about biotechnology and biosimilar. The second session of this day about regulatory requirement for biosimilar. I am pleased to announce that this session will be, the talk in this session will be about registration requirement here in Egypt about biosimilar and the biotechnology product. We have representatives from several organizations. From Egyptian Ministry of Health, we have talked about registration of biosimilar product in Egypt, Dr. Heba Ibrahim, and second talk, technical requirement for registration of biosimilar product in Egypt, Dr. Heba Khalil, and third talk will be biosimilar in Europe, regulatory body and requirement in Europe, Dr. Christoph Conrad from Paul Ehrlich Institute, and the last talk will be biosimilar market status, queue of global penetration and local specification, Dr. Gabriel Dolman. The first talk will be a regulatory overview for biosimilar and registration of biosimilar product in Egypt, and we invite Dr. Heba Ibrahim to start this talk. Thank you.
Good afternoon. On behalf of Dr. Rasha Ziada, I'm apologizing to you because she had the commitment, so she had to go. I will present her presentation on behalf of her. Sorry? Okay. That's our biological regulatory instruction on the Ministry of Health, CAPA and ORSB. They are both entities concerned of registration of biosimilar and biological product in Egypt. These are the 9 main regulatory functions, which is according to the global benchmarking tools of WHO we are concerned of. National regulatory system, marketing authorization, lot release, laboratory access, licensing of both manufacturer and scientific office in Egypt, regulatory inspection, marketing surveillance and control, vigilance and clinical trial oversight. We are a functional health authority according to the WHO organization since 2010, and we are ongoing steps for reassessments against the new benchmarking tools by WHO. Our regulatory framework overview is we are in the biological department receiving and assessment our registration file according to the CTD modules. We had a fast track procedure are guaranteed by both. We have 2 pathways for fast track. We have international price referencing is used in the pricing process of all products. And we having recognition of other NRAs and organizations, and I will mention what NRAs and organizations we are recognizing. We have Egyptian guidelines for biosimilar established since 2015, and it was published by a ministerial decree. We also have a good pharmacovigilance practice for our countries, and this is in compliance with ICH guideline, and European good pharmacovigilance practice is also applicable here in Egypt. And we follow WHO directive for GMP. These are our legal frameworks concerning the biological product in Egypt. We have 2 ministerial decrees concerning the biological or biopharmaceutical products, and we have 1 ministerial decree for the biosimilar products. The biological products in Egypt, we have 383 biological products, including 6 registered biosimilar, and we have also 12 under registration imported biosimilar product, and we have 25 under registration locally manufactured products. This is, we have 11 manufacturing sites for biological products in Egypt, and we have 1 manufacturing site for vaccines. Our achievements concerning the registration procedures, we have regulatory framework has been recently updated to simplify the registration process and to specify each step time frame, aiming to minimize the total registration and re-registration process timeline. We are also had a ministerial decree 820 for 2016, which minimized the time frame for products. And we had updates our working instruction for the products submitted for registration, whether it's registered in EMA or FDA, or submitted for registration as a CTD modules. And we had published guidance for applicant to how to give an overview how we are assessing our files. Also, we had updates our biosimilar guideline, and it's now published for public consultation. And we are inviting you to give us your feedbacks on these updates until 15 February. And we had automation registration procedure, including workflows for automation of biological product registration. And this is our EDA website. Our website has every services we are giving to the applicant, and you will find our all regulation registration step, our checklist, guidelines, our all ministerial decree are all mentioned in this in our website. Thank you. That's for Dr. Rasha presentation. That's what I'm going to talk with you. It's the registration of biosimilar product in Egypt. That's what I told you before, that we had updates our guidelines and published on our website. And we are waiting for all stakeholders to give their feedbacks on what we had update. Our key changes for this, for the previous guideline, we had introduction of new sections concerning the classes of products submitted for registration, whether it's local or imported products. We had introduced expression system and updates our rationale of a choice of the reference biological products in principle for development of biosimilar products and analytical consideration, target binding, immunochemical properties, and quality aspect sections. We had also updated sections of stability and pharmacovigilance to align with the international guideline, and we concerns about local manufacturers. So we try to facilitate it more for them to submit their requirements. And we had new subsections added in details in non-clinical and clinical studies to provide further clarity. We had restructured to give more logical flow, and we also allowed the applicant to ask for scientific advice. And this is the first time we are seeking to help the local manufacturer by giving them scientific advice. This is our classes of 4 imported products mentioned in the guideline, whether it's imported from reference or non-reference country. What we meant by reference countries, we had clinical committee decrees, which was in 2009, stating 22 reference countries which are recognized as NRAs for us. We are minimizing the step of registration for products submitted from these countries. Our local products, these are 4 classes of local products, whether it's starting manufacturing from active drug substance, or starting from an imported active drug substance, or starting manufacturing from concentrated bulk finished products, or just for filling. That's our workflow for imported products registration. Our first step is a box approval. What is meant by box approval? We had a rule by ministerial decrees that for each molecule we only have 12 products for each molecule. And after the approval of the box, we had the applicant should submit the pricing file. After finishing the pricing file, the applicant should submit all CTD file to be assessed by different parties. Different parties for all the modules of CTD. Then each concerned party should submit a report that after collection of all report, the applicant will guarantee a registration license. That's for the imported product from the reference countries. From the product imported from non-reference countries, we have a different pathway. We should first evaluate the scientific file. What we meant by scientific file is module clinical, preclinical, and pharmacovigilance data. Then, after the approval of these data, we should undergo an inspection on the manufacturing site. And after the approval of the manufacturing site, the applicant would have a box approval, and he could proceed in his registration process. Our workflow for the local manufactured biological product is slightly different, as we want to help the local applicant to be with him step by step. So he will guarantee a box approval as all products. Then he will go for pricing, for the step of pricing during 30 working days. Then the pricing certificate will guarantee him 2 years to undergo quality preclinical comparability studies and preparation of clinical protocol. Then he will get an approval for the approval to proceed in the clinical study after submission the clinical study protocol, and this phase will take 2 years. We allow him to conduct his clinical studies, and it could be extended with a justification. Then the applicant could submit the whole CTD file after finishing all his studies, and we are evaluating this file during 75 working days. Then he will guarantee the registration license. For category Z of the local product, which is a filling step, will happen here in Egypt. He will guarantee a box approval and pricing. And then he will submit the imported registration file. The imported registration file without the filling step, which will submit it after 1 year. Then we will evaluating just the remaining step, which is the filling step. Then he will guarantee the registration license. Every biosimilar product could be submitted for registration by both ways. Either the pathway mentioned in the guideline, or by the fast-track pathway, which is the Ministerial Decree 820, which shortens the timeline for registration of any submitted product. Thank you.
Many thanks for Dr. Hiba Ibrahim for this nice talk and presentation. Now we invite Dr. Hiba Khalil for talk about technical requirement for registration of biosimilar product in Egypt.
ده كليكر وده ليزر بوينتر على ده اللي بيقلب السلايد وده ليزر بوينتر اللي حضرتك عايزه تستعمليه على اوكي.
Good afternoon, everybody. It's my pleasure to be here sharing our ideas from our regulatory perspective with the industrial people for the well-being, hopefully for our well-being of our people. We're going to talk about the biosimilar guideline and the technical requirement. Of course, biosimilar guideline is a biological product. Thus, then I'm going to talk briefly about the biological product, as my colleague have said these ideas before. It is a medicinal product made of a substance extracted from, produced from a living source. Biological product differs from the chemical product. As we see in this picture, is the aspirin as a chemical product is a very small non-complex molecule. Then the interferons and the monoclonal, which is a large complex molecule. Manufacturing, briefly, I'll talk about the manufacturing process of a recombinant product. The gene of interest, the DNA containing the gene of interest will be implanted in the plasmid and then will be implanted in the host cell, whether it's a plant, bacteria, or a yeast, but usually or most probably it is a mammalian cell. Then it goes through the process of fermentation, then downgrading for the purification, ultrafiltration, and finally formulation, packaging, and finally having our final product. Influence factor during manufacturing process: expression system, vector, host cell, fermentation process, purification and modification process, pharmaceutical formulation, container closure system. Biosimilar. A biosimilar is a medicine highly similar to another biological medicine that's called the reference product. Here is this table shows the difference between a generic, then a biosimilar product. The most important things to know here, the difference in the small chemistry entity. A biosimilar is a large complex biomolecule. It is never identical to the reference product. However, a generic is. Immunogenicity is no or less regarding the generic. However, in a biosimilar, it is immunogenic. Comparison of the data required for approval of a biosimilar versus the reference medicine. As we see here, here is the reference medicine, right to the left. The clinical trial is huge regarding in comparison with the biosimilar. Then, however, the analytical part is regarding the biosimilar is larger than that of the reference product. The preclinical is minimized together with the clinical. Of course, the benefit of this change we are seeing here is the lowering the cost and lowering the time for the development of these products. What is the cornerstone of biosimilar development? As we have said in the previous slide, the comparison is mainly done through a comparability study. Development is done through a comparability study between the reference products and the biosimilar product. A comparability study, it is a head-to-head comparison regarding the quality, the safety, the efficacy, and lastly, the immunogenicity, which is a part of the safety. Comparability key element. As we said, comparability exercise, quality, preclinical, and clinical data. We'll start with the quality aspect, of course, in brief. The quality-based consideration, comparability exercise between the reference and the biosimilar products, the quality part of the CTD file is almost the same. Added to it, the comparability data between the biosimilar product and the reference product and the analytical comparisons with the reference. Comparability exercise against the reference products. Critical quality attributes that we are looking for: primary structure, higher folder structure, biological function, receptor binding, immunochemical properties, product-related substance and impurities, physiochemical properties, and lastly, stability. After finishing our quality part, then we start with the non-clinical aspect. A non-clinical study should be relative, comparative, and should be initiated before the clinical study to be done. Performed with appropriate number of batches of the reference product and the biosimilar product. In non-clinical study, there are 3 steps regarding a non-clinical study: an in vitro study, determination of the need of in vivo study. Usually you don't need the in vivo study, but maybe we could need it. And lastly, in vivo study if we determine to start it. Step 1, which is the in vitro study, it should be comparative, sensitive, and specific to provide evidence that the observed difference is in the quality attribute are not clinically relevant. It includes binding assay to the receptor antigen or enzyme, signal transduction, and functional activity. Assay should cover the whole spectrum of the pharmacological and toxicological aspect. Determination of the need for the in vivo study, as I have said, it's usually not needed. If there is relevantly quality attributes that have not been detected in the reference product, example, new post-translational modified structure, relative quantitative difference in the quality, for an example, higher impurities in the biosimilar products than the reference product, difference in the formulation, example, adding a new not widely used excipient. If we decide to do the in vivo toxicity study, in vivo study, it includes animal toxicity study, pharmacokinetic and pharmacodynamic measures, animal immunogenicity, and local tolerance. Usually there is no need to include all these regarding, then we start the clinical study. Usually we don't need the pivotal study, which is the main difference between the reference and the biosimilar products, which lowers the cost and will also decrease the time for the development of the product. The clinical study should be designed to rule out clinically relevant difference in the safety or efficacy. Factors affecting the number and types of the clinical study, complexity of the molecule, as we have said, and comparability data available, availability of a pharmacodynamic endpoint which correlate with the efficacy, for example, granulocyte colony stimulating factor, neutrophils count, safety corners with reference medicines or pharmacological class, potential for immunogenicity. As you know, in biosimilar products, immunogenicity can vary from local intolerance to anaphylactic shock. Possibility of extrapolation of other indication, which is one of the most important benefit of developing the product as a biosimilar product. Study design, it is either crossover or parallel design. Crossover is usually used for the short half-life products, less than 5 days. However, a parallel design have long half-life larger than 5 days. Dose selection, pharmacokinetic study on healthy subject, and it's usually more sensitive than the patient. If there is any change in this lowering the dose or approved dose, thus justification from the company is required. Route of administration, the same route of administration as the reference products, but if there is more than route of administration, the subcutaneous or the extravascular is the most sensitive route for detecting clinically meaningful difference between the two products. Study population, as we have said, regarding phase 1 pharmacokinetic, pharmacodynamic, it is either for healthy subject or patient. Healthy subjects, we can use healthy subject or choose healthy subject if the product is safely to be administered to them, and it is considered to be more sensitive in evaluating the product similarity. Patient, in using or choosing patient, if there is ethical consideration regarding the safety and well-being for the participant, example immunogenicity or toxicity, or if the pharmacodynamic biomarker can only be relevant in patient. We cannot see the effect except in patient. Pharmacokinetic study. It is a comparative study, should be done on to demonstrate similarity for pharmacokinetic profile of both products. Anti-drug antibody should be measured during this study. Pharmacodynamic study, it is recommended that the marker are added to the pharmacokinetic study whenever feasible. Pharmacodynamic markers should be selected on the basis of the relevant to the clinical outcome. In certain cases, comparative pharmacokinetic, pharmacodynamic study may be sufficient to demonstrate comparability study. And in this case, we can see how much the clinical trials will be minimized, lowering the cost and the time for the development of the product. You can give, again, we can see the same example. For granulocyte colony stimulating factor, the level of neutrophils in blood can be used as a pharmacodynamic marker, and it is very easily to be measured. Pharmacodynamic study is recommended to be added to the pharmacokinetic study. Pharmaco should be selected on the basis of the relevance to the clinical outcomes, as we have said. In some cases, if there is no pharmacodynamic marker, then efficacy trial will be needed. Surrogate marker, in the absence of surrogate markers, it is usually necessary to demonstrate comparative clinical efficacy. The study should be randomized, parallel, preferably double-blinded, to ensure accuracy of the results. Study design, the usual design for the studies in a bioequivalent, in a biosimilar product is the bioequivalence study, bioequivalence design. If there is any change, the company should provide us a justification for not choosing this bioequivalence equivalence design. Efficacy endpoint, the purpose of the efficacy trial is to confer comparability clinical performance of the biosimilar and the reference product. I can give an example here. Sometimes the biosimilar products have many indication. The most probable population that I can choose is not the severity, according to the severity or the predominance of the disease, but the most sensitive populations that can give me an outcome on the comparability or similarity between the biosimilar product and the reference product. The applicant should justify that the chosen model is relevant, sensitive to the detected potential difference, as I have said, with regard to safety and efficacy. Safety and immunogenicity. Immunogenicity is one of the most important concerns regarding biological product, and more certainly monoclonal. Clinical efficacy, including immunogenicity, is an important through the development program, which is usually measured during the pharmacokinetic, pharmacodynamic study, and also as a part of the pivotal study. Then it's not a separate study, it's a part of all the clinical trials done for the during the development of the product. It should include relevant patients that are not immunocompromised طبعا عشان so that I can see the effect of the immunogenicity of the drug. Clinical immunogenicity study are generally required for biological medicine, as we have said. Immunogenicity study in case of monoclonal antibodies, they are always required as it is more difficult to predict incidence of unwanted immunogenicity. And the difference, as I have said before, short-term immunogenicity, infusion-related reaction, intolerance, or long-term delayed response due to the involving immune reaction. Extrapolation. One of the great benefits of the biosimilar development of the product is extrapolation, which means if the product have more than one indication for the more than one disease is used for more than one disease, I can do my clinical trial done in one indication, and if it proves similarity between the reference product and the biosimilar product, I can extrapolate the other indication without doing an extra clinical trials. Finally, if the biosimilar comparability exercise indicates that there are relevant difference between the reference and the biosimilar product, then this product is going to be registered as a standalone development with all the clinical trial needed and the stages of the standalone drug. Thank you very much.
Many thanks for Dr. Heba Khalil for this nice and wonderful presentation and talk. The title of next talk, Biosimilar in Europe, Regulatory Bodies and Requirement in Europe. And we invite Dr. Christoph Conran for this talk. Thank you.
Yeah, thank you very much again for this nice introduction. And you see me again. First, I would like to apologize that I am not the actual biosimilar expert of our institute. It was planned that Dr. Elena Wolf-Holz would... this is the... Okay, good. Very good, thank you. Was supposed to come here. She's our expert, and she's also the chair of the Biosimilar Working Party at EMA. And unfortunately, she had a very important family commitment, so that she needed to cancel the trip, and I will do my best in order to replace her. So please accept my apologies if I'm not able to provide as many background information as she certainly would be able to do. So, I would like to talk about first the EMA a little bit, and especially the EMA commitment to advance patient-centered access to medicine. This is a very important, let's say, understanding of EMA by himself, and this is also something we embrace at the Paul-Ehrlich Institute, that the patient should be in the center, and also regarding us as partners for the industry and new developments in order to provide new medicines for the people in need in our countries. Then I would like to talk a little bit about biosimilars in the European Union, the evolution of data requirements. I guess we have heard already a little bit that the field of biomedicine and analytics is evolving very fast. And then, of course, we are dealing with some discrepant information, and I would like to show how currently the EMA or we are dealing with this. So as you might have heard or known, due to the Brexit, the EMA, which was located in London, had to move from London, and that already started in March last year, to Amsterdam, first in a building in between, and now finally I heard from colleagues they already had their first sessions in the new building in Amsterdam of the EMA. And of course, this was a huge transition and unfortunately also loss of some personnel. And the way the agency was trying to cope with this challenge was that the business continuity was granted for all the key features like granting marketing authorization, providing scientific advice, and so on and so on. And the activity they were kind of slowing down a little bit was developing new guidelines or revisiting guidelines, and this is something they, of course, need to catch up. And this is something which is also very important in order to see how EMA is trying to promote and support development of biosimilars and new medicines to the patients. And as already mentioned, we have very fast and big changes in the field of medicine. New technologies are coming up, analytical technologies are getting better. And so the EMA started already in 2018 to think about an EMA regulatory science strategy. And this strategy led up with a lot of contributions from 150 different stakeholders or people supporting that, and this is a strategy towards 2025. And part of the strategies are 5 goals: dealing with science, dealing with access to medicine, and when we look, this is goal 3, to access to medicine, which they summarize here as the goal, advancing patient-centered access to medicine in partnership with healthcare systems. You see here that promoting biosimilars in healthcare system is also one strategic goal and activity the EMA will focus on in the next couple of years. And if we look into our law in order to understand how EMA is dealing with biosimilars, we actually have a definition of biosimilars, and as far as I know, this kind of definition for biosimilars is pretty unique worldwide, basically in our law. And it makes a differentiation between generic medicinal products, where we talk about the same quantitative and qualitative composition, and when we talk about biosimilars, we are talking about similar, as the word is already saying, a similar product with regard to a reference biological product. And this is very important to understand. The work of regulators, of course, is always based on the current knowledge of science and technology, but regulators also have to work within their legal framework and develop the way they assess and register products within this framework further by using and recognizing science and technology. When we look at the market or the situation of biosimilars in Europe, you see that since it has been started in Europe, already 92 marketing authorizations have been submitted. 80 have already been reviewed. 12 are currently under review. This is the state of 10th of September 2019. From those who have been reviewed, 2 were negative, 61 with a positive opinion, and 17 actually have been withdrawn by the manufacturing by themselves, and this is even something which happened post-approval, awaiting the EC decision, that these kind of withdrawals happened, which for us as regulators is difficult always to see and understand and know what is the background for that. And if we look at the type or what kind of biosimilars are applied for and approved in the European Union, we talk about these 63 different brand names or 45 approved products, which are referring to only 15 different reference products. So this is the kind of market in Europe we are currently talking about, and the whole list, of course, we don't need to go into detail, is here on the left-hand side, where you see the different type of biosimilars referring to the 15 different reference product, just to give you an oversight about the magnitude of products we are talking here. So this slide has been shown already at least 2 times, if I remember correctly, in a different way, with this connotation of the world is turned upside down in order to illustrate that different amount of data is used or needed for biosimilars. And you might argue with me whether this pyramid, where the data seems to be the same for the reference medicine in terms of quantity and the biosimilar medicine, and we can debate that, and we will see in the examples we will come to that this indeed needs to be discussed, how big each of these portions indeed should be. But in principle, you see the comparison. Classically, a medicinal product is based on the quality, non-clinical studies, and then the huge amount of clinical studies starting from safety, starting from immunogenicity, PK, PD studies, and also then the big safety and efficacy study. And then, of course, in the end, we also have the risk management plan in order to make sure that the safety through marketing is granted. When we go to the biosimilar side, we also have, of course, as a basis, the quality studies. And I think we all agree that this has to become more and more relevant, and in the kind of diagram the previous presenters showed, it was clearly stated already that this for biosimilars is the majority of the data. Then we have also comparable quality studies, and this makes this quality portion, of course, already bigger. Comparative non-clinical studies, we also need to look into, and then again, comparative clinical studies to the magnitude they are asked for, depending on the data which is provided with regard to quality and non-clinical. And then the risk management plan is pretty much the same. And this is taking into account what I already mentioned. We have a tremendous development in analytics and in other scientific fields of medicinal. Oops. Is it me? No, okay. And this is, of course, something we need to take into consideration in the future regulation of biomedicines. And you see here just given an example about mass spectrometry, where you have a 10,000-fold increase in sensitivity already during the last 10 years, and even more during the last 20 years. And the kind of new paradigm which has been introduced here or is discussed is that we are looking and asking for a confirmation of sufficient likeness, similarity of the product. And let's look what this could meant to be. Keeping this in mind and taking into consideration this kind of development, the EMA started a pilot project on biosimilar scientific advice. And the idea is to look very closely into, they were saying we need to have at least 6 scientific advice procedures we look very detailed into in order to see whether we can give early on scientific advice on the quality data, and then based on the quality data which is available, make a tailor-made plan of what kind of comparability data in preclinical or clinical settings are needed. So far, this has not been concluded. 4 procedures have been completed and analyzed. One has already been started. It is expected, but maybe that will be a little bit delayed, that towards the end of this month, this pilot has been finalized. But there are already a couple of experiences which could be made, and the problem which was faced, and manufacturers might confirm that here, that this concept of stepwise approach is very often quite difficult, since time is really money in this business, as with other business as well. And the development of a new medicinal product with regard to the development of quality, non-clinical and clinical, is very often going in parallel. So this kind of approach to go stepwise from quality towards clinical seems to be a little bit difficult. So when we look at this table, then you see on the right-hand side the current clinical study recommendations from different products, and they are listed according to their complexity. This is something we also had quite often now in the previous presentations, that of course biosimilars, if you're looking to insulin up to monoclonals, they are quite different in their complexity. And you see that this is kind of reflected also in the recommendation of clinical studies, going from the classical PK/PD studies in human volunteers, but no efficacy, no safety trial, with a less complex product, down to PK studies in one therapeutic area, plus efficacy safety trial, plus PK data in another therapeutic area for monoclonal antibodies. But this is under discussion. And taking into consideration what I showed previously with the advancement of technology, the ones which are here in yellow boxes, and I guess this is the kind of approach we as regulators, and I hope you agree, need to move forward and maybe need to accelerate even a little bit. These are the kind of products where we can certainly start discussing, can we go with quality data only, and are we able with all these technologies to show this kind of similarity that we don't need these confirmatory clinical studies. And this is indeed under discussion. And let's see how EMA is going to move forward. I'm not the expert to predict how fast and soon all these changes will be implemented. But the way how EMA is looking into the similarity and the safety of data we have for the licensing of biosimilars will be shown now in the way how EMA is dealing with discrepant information which are out there here and there with regard to biosimilar medicinal products. Maybe going back. So I would show you an example about pharmacovigilance and traceability. And then it will be an example about combination therapy and the possibility for that. And of course the interchangeability of biosimilars and what is the position of EMA in this regard. And there has been a project to look whether there has been a change in the in the way to identify biosimilars in the market in our pharmacovigilance system. And if you look here at these data, you see that with these two examples, infliximab and insulin, there hasn't been that much of a change whether the biosimilar or the reference product was able to be identified in the pharmacovigilance system with regard to the period before the biosimilar actually has been introduced. So on the far left side, you see there was no biosimilar, and you have a small amount of percentage of cases which were not be able to identify, and this kind of small amount actually stayed pretty much the same even with the introduction of biosimilars, and this is kind of promising the situation in pharmacovigilance and the data we can generate out of that. And the same actually is shown here for insulin. The way these data have been generated is through our pharmacovigilance system, and then the reports were extracted manually out of the ultravigilance system and were reviewed manually in order to identify whether the manufacturer was able to be identified or not. On the other side, and that makes the discussion not that easy as it looks like, is that there were also studies which show that the European approach might not be that robust as it has been shown in this study, where people were showing that, for example, nearly 26% of infliximab safety reports in Europe did not include brand names, so the identification was difficult. What it doesn't show here is whether maybe batch numbers have been mentioned, which would then also be allowed to link to the actual brand names, but these are details in this study. And another evaluation showed that 59% of drug reaction for trastuzumab were reported only by non-proprietary names. And taking this together and comparing to the data before, this is still something showing that the pharmacovigilance system, of course, needs to be robust in order to make sure that our approaches are working and that we know how different products are actually working on the market. Another example of discrepant information EMA or regulators in Europe had to deal with was that a company, for example, stated that its particular medicine cannot be prescribed in combination with a particular biosimilar medicine. So that was an initiative coming from an originator manufacturer. And then the agency we're looking very closely into, okay, what is our position with regard to combination therapies? And this is stated later that a particular medicine is authorized for combination treatment with any medicine with particular INN, and this is up to the healthcare professional to decide which particular INN medicine to prescribe, taking into account national legislation, prescribing practice, and any relevant guidelines. The situation here is different. FDA is taking a different approach than EMA. This might be due to the fact that, of course, Europe, with all the different national countries and their own legislation, we have to be there a little bit more flexible and cannot just make one strategy for all Europe, as the FDA is very often able to do. So here the approach of the EMA is that in the end, the responsibility is given up to the healthcare professional within his jurisdiction, but in principle, it is clear that combination therapy with biosimilars is, of course, possible and should be decided within this framework from the respective people. Another topic is addressing the interchangeability, and there was a company's patient brochure of an originator emphasizing that a corresponding biosimilar is not interchangeable and implies that it may not be as safe or have the same result. And even a YouTube video from this company was kind of implying that the biosimilar is unsafe. None of these informations about safety or clinical performance are true and rather undermine efforts to promote the acceptance of biosimilars. And when we look into this topic of interchangeability, there have been a lot of studies around. One example I just gave is spreading a little bit of doubt, but a lot of scientific papers have been published over the last couple of years, and I'm just showing them here. This is one part where I guess my colleague would be able now to summarize and give some details for each of them, but I'm sorry, I'm not able to do that. But in the end, and I guess this is something really worth looking into in order to understand the current understanding in Europe, is a publication then where also our expert is part of Elena Wolfholz, summarizing our position with regard of interchangeability and with the summary that we do think that switching is acceptable and post-approval in this context monitoring should be suggested. So it is understood from the EMA, and that I was already given very much at the beginning, so there's a strategy at the EMA to promote and support the development of biosimilars, and it has been shown that we need to work on several areas in order to make sure that it is properly understood, and this is indicated in this slide, that of course we need to strengthen the dialogue with all the stakeholders. And this has already been started with the European Commission with a multi-stakeholder conference in September 2018. And there is a need for an increased collaboration with healthcare practitioners. This is certainly important in order to educate and to promote consistent messages coming from regulators for our healthcare practitioners and also for patients out there being in need of the medicinal product. Another part where we certainly, and I will give you then towards the end of my talk an example, all the regulatory agencies are also in need in updating, for example, their web pages in order to provide up-to-date and scientifically sound information to healthcare practitioners as well and the patients. And of course, with our pharmacovigilance and all other monitoring systems, there's a need, and it will build trust to continue to be very transparent with all kind of benefit risk, which we can observe and we know about biosimilars. Just to give one example of the need, which is still also in Europe in order to disseminate information about biosimilars, and that almost half of the national control authorities' websites in Europe contain almost no information about biosimilars. And stakeholders recognize the role of EMA in guiding and informing them about biosimilars, but there's room for improvement about the guidance for NCAs about the use of biosimilars in clinical practice. And this gap seemed to be partially compensated by information from healthcare professional associations, but this is of course not sufficient, and every stakeholder in this field has his responsibility. And with this, I would like to thank you for your attention. This is not Germany, this is Helsinki, because the slides were compiled during the presidency of Finland, and this is giving regards to our colleagues in Finland, and thank you very much again for your attention.
Many thanks to Dr. Conrad from Powell Ehrlich Institute for this nice talk and presentation. The title of the next talk, Biosimilar Market, Status Quo of Global Penetration and Local Specification. Now we invite Dr. Gabriel Dolman from Biopharma Excellence for this nice talk.
Thank you so much, and good afternoon. It's a pleasure to be here and the pleasure to close the day of very, very interesting contributions, a broad variety actually, and I was asked today to give you some insights and overviews on how other regions regulate biosimilars, and also to give you some market numbers. And I think I have also found some interesting numbers to further confirm the main conclusions you gave already today, that the biosimilar approach is an important tool to save costs in health systems. I can say it is a very interesting discussion because I can remember very well being part of the team who has approved the very first monoclonal antibody for therapeutic use, which is quite a while ago. I was the rapporteur for Herceptin in Europe, trastuzumab. It was a very challenging approval at that time. I was the co-rapporteur for Remicade, infliximab, and now we talk about the follow-on products to these blockbuster products, which are not only interesting from a commercial point of view, they have changed treatment paradigms. They have offered treatment modalities to patients which were simply not there before they came into the field. It is oncology, it is autoimmune disease, it's rheumatoid arthritis. And that shows us the importance of authorities, the responsibility they have to take in order to ensure that any follow-on product is as efficacious and safe as the originator. On the other hand, we have to ensure that we save costs in the health systems in order to release budget for future innovation and to allow access to patients. So I think this discussion is extremely important, and I don't touch on European requirements in that talk here, because this will be covered tomorrow in a more extensive contribution. So today it is regional definitions of the regions, then Europe, the local specifications, and then some numbers on the market. When you look around the world, you will see that many regions and many countries have meanwhile introduced the concept. But you need to understand what is the legal basis when you discover and develop a new biosimilar for a specific region. And of course, you see there are many different definitions which are all meaning the same. It is a follower product to a reference product, originator product, and again, we also have here different definitions per region, per country. The underlying principle, as we see, is the analytical similarity is the basis of the pyramid, as we have seen. We need evidence that there is no clinically meaningful difference, and at the end you have a product in your hand which can be used as the originator. I would like to mention that the point on the reference product is also legally defined in each jurisdiction. It is EMA, it is Europe, it's Japan, it is US. You have in the law the necessity that your originator product has to be approved in that country. And I think that makes sense, because this is the one which is used in the health system. So therefore, originally there was a very clear requirement that the full development has to take this originator product only. But meanwhile, and this is also a very important aspect in that field, authorities progress with the knowledge, they progress with experience. And meanwhile, all these three regions allow the use of a reference product of another region under certain circumstances. And that is a very big relief, because reference products are not easily to source, and specifically for the clinical development there is huge amount necessary, and so companies can source from regions where it is easier. And you saw this pyramid, and I really want to make that point extremely strongly. For the biosimilar, it is the process. It is the analytics which really have to show that you have a product in your hand which is highly similar. What should happen if you have overlapping curves? What should happen if you have the same amino acid sequence, a highly similar glycosylation for antibodies, a similar functionality, etc. with the product? It will work the same. So therefore, this is a very important basis. And only in cases where you don't fulfill this paradigm, you will also see differences in clinical means. The extrapolation was already mentioned, and this is an aspect which is, of course, the underlying concept of the biosimilarity. You simply don't need to repeat the pivotal studies. You can really extrapolate from one fundamental package to all indications. And that makes the development abbreviated, finally, and that makes the product interesting for the health system, because it costs simply less money. Going a bit deeper into the details, the US FDA has established the legal basis, and this is the only region so far, to my knowledge, that has a legal definition of an interchangeable product. And we should really be careful with that term, because the interchangeability is something special. In the first instance, every product has to be approved as a biosimilar. And only if the company develops the product as an interchangeable product, which means the pharmacist can substitute the product without prescription, without involvement of the prescriber. But for that, there must be a very specifically designed clinical study. Can you imagine to investigate two products in a crossover study by which you switch the product after 6 treatments into the other arm and do the same for both products? This is an extremely difficult study. It takes very long time, and at the end you have to show that the product works the same in each patient as compared to the other product in the same patient. This is not trivial. It is a very special category, and therefore, so far, there is no interchangeable product approved in the US. There are a couple of studies ongoing, but it's not yet arriving in the market. FDA has a lot of guidelines, and very recently they were updated specifically in the quality section, but also in the clinical section. And I would like to mention at that occasion, agencies can learn. Regulations are not carved in stone. They change, they amend, they modify, because in the moment they learn, they can, for example, withdraw a guideline, and that happened in the US with the guideline on the statistical considerations of the biosimilarity development, because this was simply blocking development, because that required a huge number of products in the batches in the development. So they withdrew that and have replaced it meanwhile by more modern considerations in the quality guideline. And another interesting example is this clinical immunogenicity considerations for insulin products, because it says there is no need to investigate clinical immunogenicity for biosimilar insulins. And that is very new, and that says simply we know that all insulins induce immunogenicity, and we know that insulin works despite that, and we know that we can change insulins. So why should we then ask companies to do that immunogenicity assessment in the biosimilarity assessment? So there are really a lot of there's information out which are considered by agencies, and they learn. And we have also heard before that there is meanwhile agreement in the FDA and also in the EMA that certain products can be approved based on healthy volunteer studies and PD markers such as filgrastim. That was the case in both regions. FDA, as Europe, follow a stepwise approach. I think I don't go into that in a lot of details. We have heard that already. And this is what I mentioned before, the number of lots needed to develop a biosimilar is quite high, although we don't follow anymore this very restrictive statistical approach. But you can see here that is a typical number for reference product, 10 lots. For the analytical similarity, for the biosimilar, it goes into that direction. And if a non-US reference product is used, then it is necessary to perform a pairwise comparison first analytically before the FDA non-US reference product is continued in the development. Risk assessment. I mean, you will never have the same development package in two different products, even if it is the same product class. You always have a very individual development. For that reason, agencies, not only the FDA, want to see from you, from the developer, that you understand your product, you understand your process, you understand why you do in certain areas more than in the others. So this is this famous risk assessment tool. And so all data you have will have to be discussed in terms of what does it mean to the clinical performance of the product. Do I have to go back into the process before I continue into clinic, or can I go into clinic without further fine tweaking of the process? The tier concept, so what parameter of an investigation is critical? You develop these kinds of 3 tiers. The most critical is normally tested for equivalence, tier 1. Tier 2 is normally tested by smaller differences allowed. And then the 3rd tier is normally just visually described. Actually, FDA went away. They don't anymore like to use this tier concept. They want to see a gradual risk assessment with criticality ranking. But the underlying principle is, I think, still the same. Moving away from FDA, short comment on Japan. Japan already is also very active in that field. And that's an agency which has published a guideline, and originally they have requested that only Japanese reference product is used in the total development. Meanwhile, they are also moving away. They allow other regions reference product. And one specific I would like to mention is that still they require a certain number of Japanese subjects which need to be involved in the clinical program. So this is, I think, special, but I think not only for biosimilars, this is the same for any other product to be approved in Japan, so the impact on the ethnicity. So that is something a company has to understand. The rest in the program is relatively comparable to the European and the US requirements. WHO was mentioned. WHO plays, of course, a very special role in the world to ensure the access to medicines in emerging countries. They have had a guideline already quite a while, and I would like to mention one point that just recently, end of last year, WHO has pre-qualified the first biotherapeutic trastuzumab based on a biosimilar which came from Samsung. And this was based on an assessment which was originally done by the European Medicines Agency, but the WHO has put on top their own qualification criteria in order to allow the distribution of that product in many, many countries where normally no harmonized tool to assess products like this are existing. I think that is a big success, and we know that there are other initiatives underway at the WHO level, such as find one company that has a product, shares the process with many other companies in other regions which produce the product following that process, and then there is one package of data which allows the product to be marketed in many countries. I think that is a very useful initiative. And with this pre-qualification medicine scheme, here we have the numbers they gave on the website end of the year, so they really can significantly reduce the price, and which is already the case now, and after the pre-qualification they plan or expect that it's even decreased further. India is, of course, in region to a country to understand a big market. Many patients, they have the guideline on place quite a while as well. They have some specific requirements mentioned. Principally on paper, everything is comparable or a lot comparable to ICH and European and FDA requirements. However, as far as it is understood, the clinical package is normally significantly smaller than we see in Europe and in Europe. It is not fully understood what the final clinical package is, but it is significantly smaller. Latin America, there are a couple of countries. It is Brazil, it's Argentina, it's Mexico. They have their own systems in place. And also Brazil has a system in place which allows the assessment based on the origin of the product. Is it an own country product or is it an imported product, for example? And they have fairly comparable requirements to the European and FDA system. Mexico, I mentioned that to participants in the break. My impression was that Mexico was originally quite active, then there was big silence, and meanwhile there is a new, I think, renaissance, so there is a guideline out, but at the end, so far, not many products following that system. We will see what is the case with Mexico. Argentina has had a very strong sector in that field, and I know from direct involvement also that the pyramid of the data requirements is clearly based on the analytical and process comparability, whereas the clinical similarity package is slimmer than we know it in Europe and the US. Normally there is a data package of 100 to 200 subjects, which is sufficient for approval. China has been now developed into an agency that has the requirements published. They have also taken their time to learn from European and the US agencies. They have developed guidelines. They also have practical testing as part of their assessment. They also require a stepwise approach for the comparability. However, so far, although the guideline is out, it is not 100% sure what is the final clinical evidence. We know that global developments which go to China always are required to include, again, a small population of Chinese subjects. So this is a very specific requirement, as in Japan, also for China. Russia has also changed their requirements. They originally started with a program that allowed the biosimilar to be developed simply based on the comparison to the monograph. There was no comparison to a reference product. They took the monograph parameters to which the product was compared, and then there was a small study of 150 patients or subjects. That was it. Meanwhile, it has changed. The guideline is not anymore out, and it seems to be important that there is a good interaction with the agency to develop an individual program. With these examples, very quickly summarizing the individual requirements, I would like to summarize that for regions outside US or Europe, there are very individual requirements. What is in common is the fact that there is clearly an agreement that the generic approach would not apply to any biological. they all work with the term reference product to be used as the comparator to which the biosimilar has to be compared. The details which are relevant for the non-clinical and clinical studies are often not given in the guidelines. And indeed, there are big differences between the countries. I would say there is a focus on the analytical similarity evidence, and normally there is then a small clinical study performed, not with a focus on equivalence. And this was, I think, the summary for all countries, excluding FDA. Of course, the FDA in the US requires clinical studies with the purpose to show equivalence. With this excursus into other regions, I would like to close the session with some numbers and getting also confirmation for what was discussed before. We saw numbers already on approved products. And I think Europe plays a revolutionary role here in that field, definitely. They started that concept. It came with Omnitrope, the first biosimilar ever, on the table of an agency. The scientific data was accepted, but there was no legal basis. And that was the founding point of that legal article in the European drug law, which since then allows the approval of the biosimilar products. And you can see here that we have many approved biosimilars by trade name and a lot of different active ingredients. 16 did we count. And I would like to mention there are, of course, withdrawals to many different reasons. And there were also some rejections, negative decisions. But these rejections, they were all caused, specifically the very recent one, on other reasons than pivotal study data reasons. It was a GMP failure, it was a PK study failure, but normally no CMC data failure, and definitely not a phase 3 study failure. Although they are all designed for equivalence, none of them has yet led to a rejection. US, they have a little bit less products approved yet. They are not all on the market. As we know, various European approved products, they are on the market. So we have 9 different active substances and around 30 biosimilars by trade name. Japan, interestingly, has also quite a number already approved. 10 different actives and 18 approved products by trade name. Now, what do we know for the market penetration of biosimilars? I think there are many factors which influence that, and it will be different per region. In Germany, for example, we have so many different health insurance companies, the north, the south, the east and west. They have different agreements, criteria. It is not a one country approach to agree on the price, for example. That will influence the market penetration of biosimilars. However, there is not only a big political will in Europe to get the biosimilars on the market, there's of course also the cost pressure in the health system, so they will come. In other regions, such in the US, it is certainly different because there is this so-called patent dance to go through after the product has been approved. So it will not automatically come to the market. When we look at this number here, which shows us on top the global biologics market, and here we have the global growth as this curve here, you see the global growth is double digit until 2018. Now it's a bit slower here. And we see the blue is US, the green is Europe, the orange is Japan and other regions. So you see how that is evolving over the years. And this is the revenue number in US dollar in billion. This is this number here, and that is the annual growth per region in percentage. The same figure we have for biosimilars here. So that's US, that's Europe, that's Japan. That's the absolute number of the revenue, and this is the growth rate here. The growth rate is, of course, huge as compared to the biological market, but of course, in absolute number, much smaller. On the other hand, I would like to comment one point. We have many, many players in the biosimilar field in the world. As we have heard, there are 50 companies maybe who develop one and the same monoclonal at the moment. But they reach a certain stage. They reach a certain preclinical stage. They have then to partner with someone who can afford the development into the clinic. And therefore, the number of players or the number of products under development does not mean that they will all reach the market. So this step into the clinic, into approval, is something which requires a lot of efforts. The global sales estimation that comes here, so we have in blue the biosimilar market, and then as compared to that, the off-patent biologics market. Of course, these are all prospective numbers, but that shows when we look at the total sales in billions, that there is a very optimistic outlook. Interesting number is given here, of course a bit unfair against China as the first box here. This is of course a huge market, and we have here the market size 2018 in $1 billion versus prospective outlook into 2025 from 2 to 8 billion. This is the annual increase in the market size from in 2025, and you see other countries with the same color code. So I think that is a very, very interesting outlook. Every country, every region has the same pattern increase in the biosimilar market penetration. One interesting example will close my talk. I got that just end of the year from the Handelsblatt, which is a business journal coming each day. And that was on Humira in Germany. And that was on the market share we have in September 2019. And you see Humira, that's the originator, 50%, only 50%. So that is really interesting number. And then we have Biogen Imraldi with 15, Amgen's Amgevita with 15, Hexal, Sandoz product 11, Mylan's 8, and Fresenius Kabi, who just got the approval recently, was less than 1%. Okay, that's the market share, but interesting fact is the following, that Biogen has obviously significantly reduced the price very, very recently, Humira is now almost twice as expensive as that product. And that has induced a kind of price war, because all the other players here, of course, have to follow Biogen in terms of price reduction, so that the market participants will, including Sandoz and Fresenius, become under pressure and will further reduce the price. So you know that Biogen product is developed together with Samsung, so that is Samsung's biosimilar Humira. And here we have some numbers also for the German market for this product. And I would like to mention that fact here. 1 year before Humira patent expiry, there were 66,000 patients treated. 1 year after Humira patent expiry, you see the number increased already to close to 80,000. So there is a lot of patients also, of course, in a country like Germany who don't get the product. Now we can make that available to those patients. So after this first year of patent expiry, Humira already gave up 50% of the market. The originator has only 50% of the market, and I think it will be even more because more is on the horizon. And then on top, for the 50% shares for biosimilars, the price will significantly be reduced over the next time. So with that, I summarize this session here, and I think this concept of biosimilars is highly interesting, a politically supported concept. It requires a well-established legal and scientific basis for the abbreviated development in order to ensure the product is as safe and efficacious as your originator. It saves costs in the health systems. It allows access of more patients to medicines they need, and it releases budget for use for new medicines to serve unmet medical needs. Thank you so much.
Thank you for your nice presentation, and we hope that we get a nice day and get good information about biopharmaceuticals and biosimilars. Now it's time for lunch, so we have it's opened now. You can get the lunch now, and we hope that we can get nice lunch. That's